Hi folks, my name is Ethan Moyer, and today I'm going to be talking about my STAR project with Professor Khan titled Measuring Protein Structure Deviation Using Machine Learning. This project focuses on a long-standing problem in bioinformatics called the protein folding problem, which has many applications in biomedical engineering, including drug design, protein engineering, and the functional annotation of proteins. The gist of this problem is given a simple amino acid sequence, can one generate its complex three-dimensional structure. Now, previous methods focused on an energy minimization problem, where given an amino acid sequence, they generate decoy structures, or non-optimally folded proteins. By changing these decoy structures in such a way that the energy decreases toward a minimum value, they're able to generate native structures, or optimally folded proteins. This method is shown in the motivation figure, where with energy on the y-axis, each decoy structure has a much higher energy than its native structure. A more optimal approach is to instead measure the energy deviation of each decoy structure with respect to its native structure. This approach is shown by the red to blue gradient in that same figure. And this deviation is what this project attempts to measure. Our first step was to create a decoy dataset of proteins. This is done by identifying appropriate native structures and randomly shifting all of their atomic positions slightly. In order to predict metrics on each decoy dataset, each sample needs to be represented in an intelligent way. One method is to encode each structural protein into a 3D grid-like representation where each 1 by 1 by 1 angstrom unit holds information about a single atom. Not only does each atomic unit contain features that describe an atom, but it intrinsically contains information about where each atom is located in the protein. For this 3D grid representation, 3 features are assigned to each atomic cube. The first feature is a label for the generic atom type, so whether it's a carbon, nitrogen, sulfur, or oxygen atom. The second feature is for the positional atom type. So for instance, whether a carbon atom is an alpha carbon or a beta carbon. The final feature is a value corresponding to the occupancy of the nearest atom for each atomic cube. A second protein representation is simply the chain of amino acids. This 1D representation just has one feature for each amino acid in the protein. For the 3D representation, there are 3 different metrics that describe the energy and structural deviation for a decoy protein. The first metric is from an all-atom scoring function from Rosetta, which is a computational and structural modeling software package. The second metric is a contact map of a decoy protein. All a contact map is a n by n matrix of pairwise distances between atoms in a 3D structure. The third metric is the mean squared error of a decoy contact map with respect to a native contact map. For the 1D representation, there's just one metric. This metric is the root mean squared distance of 2 protein structures reconstructed from contact maps using a method known as multi-dimensional scaling. All of these metrics were predicted using their own respective convolution neural network architectures, as shown at the bottom of the methods section. For the 3D network evaluation, we fed a dataset of 1,000 similar decoy structures that are 10 amino acids long into 3 different convolution neural networks. The mean squared error loss for each network converges, which indicates that our model is able to at least learn its metric to some degree. For the 1D network evaluation, a different decoy dataset was selected from a list of different protein structures. The converged root mean squared distance of varying dataset sizes was graphed for both a convolution neural network and a traditional one-layer artificial neural network. The proteins in the application section were reconstructed using multi-dimensional scaling of the predicted contact map and the actual contact map from a dataset of 100 proteins using the artificial neural network. The left prediction is the best case scenario with a root mean squared distance of 0.92 angstroms, and the right prediction is the worst case scenario with a root mean squared distance of 4.347 angstroms. The average root mean squared distance for this dataset is 2.136 angstroms. These demo predictions are a direct application of this project because although the best case scenario had a root mean squared distance below 1 angstrom, we would need our prediction models to be far more accurate than that. In the future, I plan to compare energy minimization to the 3D metrics described in this work with the hope of achieving much more accurate results than those generated from the one-dimensional network. Also, I would like to mention that we have recently submitted this work to as an abstract to the 2020 Mid-Atlantic Bioinformatics Conference hosted by the Children's Hospital of Philadelphia's Department of Biomedical and Health Informatics and the Institute of Biomedical Informatics at the Perelman School of Medicine at the University of Pennsylvania. Thank you for listening.